Hello everyone and welcome to Instant Biology by Dr. Nilab. The topic that we are going to start today is Introduction to Plasmids. Now you would be knowing that we are already running a playlist on basics of RDT, that is recombinant DNA technology, and this Introduction to Plasmid video is just an addition to that basics of RDT playlist. Okay, so this plasmid topic is a very long topic. I would be making multiple videos on that, and today's video is just an introduction to the plasmid chapter or topic. So before going into the real details, first of all let us start with a very small question, that is, what are plasmids? If someone asks you, can you define a plasmid? Yes, of course we can define it. So what do we understand by plasmids? Plasmids can be considered as most often circular double-stranded DNA, and they are found inside the cells. Now let us understand each and every sentence clearly. So when I say they are most often, what does most often means? It means generally. That means there are other characteristics of the plasmid also. Plasmids might have other characteristics as well, but this is the most general characteristic of plasmid, that is they are circular. When I say circular, what does it mean? So circular simply means that it will have this kind of a shape, okay, circular. Plus there is also one word, that is double-stranded DNA. You all know what DNA is, but if I say double-stranded DNA in a circular fashion, it would be something like this. This is how we can represent a circular double-stranded DNA, and it is found inside the cells. Now when I say cells, I am referring to mostly bacteria. I am mostly referring to bacteria. Why? Because these plasmids are naturally occurring inside bacteria. You do not introduce external plasmids inside bacteria, they are naturally present inside the bacteria. So whenever I am referring to plasmid, more or less it is depicting a bacterial cell. It is present inside the bacterial cell only. Now the next statement, let us understand this. It is not attached or associated with the chromosomal DNA. What does that mean? It is not attached or associated with the chromosomal DNA. What does that mean? So you would be knowing that the chromosomal DNA inside the bacteria is a separate entity, okay, whereas the plasmid, as I have already said, it is a separate entity. If I take the example, now look at this image, you can see that this is the bacterial DNA, right? You can see that this red colored, or the DNA that is depicted in the red, it is the bacterial DNA, that is the genome or the chromosomal DNA of the bacteria. But if I talk about this blue colored DNA, this is representing plasmid. Now you can clearly see that both of them are not associated. That means bacterial DNA is not at all associated with the plasmid. Both of them are present separately, okay. That is why we have written that they are not attached or associated. Now this is not always true. They can be associated, but remember we are talking about the general conditions first. We would be talking about the specific conditions, but in a later lecture. Okay. Now the next thing is, it can be considered as a true replicon. What do you understand by a replicon? Replicon means one that can perform replication. One that can perform replication by its own, okay. They do not need help. They can perform replication by their own. Now you would ask me that, sir, how can a plasmid perform replication by its own? What characteristic allows the plasmid to do so? Now always remember, and I would be talking about this in detail also in the upcoming lectures, that this plasmid, it is having one unique feature, that is origin of replication. Origin of replication, what does that mean? Because origin of replication is present,
origin of replication actually allows the plasmid to replicate itself or to make its own copy. Now something that is able to make its own copy can be called as a replicon. That is why plasmid is considered as a true replicon. I believe you would have understood this. Now coming to the next point, the plasmid can be stably inherited in an extra chromosomal state. What does that mean? Now suppose I'm showing you that this is one bacteria. Now this bacteria divides into two daughter bacteria. Okay, this bacteria divides into two daughter bacteria, suppose. Now suppose this the parent bacteria had one plasmid like this. Now this particular plasmid can be inherited by the two daughter bacteria stably. That means there would be no breakdown or destruction of the plasmid during the process of inheritance. It can be stably inherited in an extra chromosomal state. Now what does this extra chromosomal state? Now this would be possible. Extra chromosomal means outside the chromosomal structure or outside the bacteria's real DNA, it can be stably inherited. Okay, so this is the condition where we are saying this is actually the extra chromosomal condition being depicted. Extra chromosomal. Why? Because the plasmid is present as an extra chromosome. It is not attached to the DNA. So whenever it is in extra chromosomal state and the bacteria divides, it can be stably inherited by the daughter cells. This is what I am trying to say. The next thing we have already talked about. I told you in the first point only that these plasmids can be naturally occurring inside the bacteria. Whenever I talk about plasmids, it is in general we are talking about bacteria only, because plasmids are present naturally in bacteria. You do not need to insert any foreign plasmid. You can, but it is present naturally inside the bacteria. I hope you got that. Now, having said that it is naturally occurring in bacteria, please do not think that eukaryotes will not have any plasmids. Okay, eukaryotes also have been reported to have plasmids. And what is the example of that? The example can be yeast. Okay, let me just introduce you to this. So you might have heard about yeast. It is basically a fungi and it has 2 micrometer plasmid. Now initially I told you that it is the bacteria have naturally occurring plasmid, but in eukaryotes also plasmid has been seen, has been observed, and the example is yeast. The plasmid is of 2 micrometer size, 2 micron size. Now anything that is present inside any cell would definitely have some functions. Why would a plasmid be present inside a bacteria? There should be some function associated with the plasmids. However, we would be talking about the functions in depth, but for now I just want to tell you about little bit about the functions. So the first function is antibiotic resistance. Antibiotic resistance. That means that one bacteria that is having a plasmid, let us say that this is one bacteria and that is having some plasmid, it is having the antibiotic resistance plasmid present. Now it can transfer, this antibiotic resistance plasmid can be transferred with the horizontal basically horizontal gene transfer. Through the horizontal gene transfer it can be inserted inside any other bacteria. Now previously this bacteria was only antibiotic resistant, but now this bacteria is also becomes antibiotic resistance. The next example is, I would be talking about this in detail. The only thing that I want you to understand is that the plasmid can be used to transfer resistance from one bacteria to another bacteria against an antibiotic. Okay. The next thing is colicine resistance. What is this colicine? Colicine is basically a toxin. If this colicine resistance gene is present, the bacteria will become colicine resistant, And just like the antibiotic resistance gene, which is being transferred from one bacteria to another bacteria through the plasmid, this colicin resistance can also be transferred through plasmid. Other features or other examples include symbiosis. Okay, that means mutualism. Next example is nitrogen fixation. The final example is tumor induction in plants. Now you would be knowing about Agrobacterium tumefaciens, right? It causes or induces tumors inside plants. How come it is inducing? Because of the Ti plasmid that is present inside the Agrobacterium. For those of you who do not know about this, they can just browse through my playlist. There is a video on Agrobacterium mediated transformation. You can study that and it will give you a very clear picture. Okay, so these were some of the functions that bacterial plasmids confer upon the bacteria in which they are present. I hope this is clear. Now let us move on to the next point, that having so many advantages or having so many properties, please do not ever think that the plasmids are indispensable for bacteria. What does that mean? Do not ever think that plasmids, without plasmids, the bacterial cells cannot survive. This is not true. Bacterial cells can easily survive without plasmids. Bacterial cells can easily survive without plasmids. So why are we studying plasmids? What importance does plasmid hold? Plasmids just provide an added advantage for growth. That means if a plasmid is present, then it would be having more advantages in terms of growth in comparison to the bacteria that is not having a plasmid. Okay, up till here I believe everything would be clear. Now coming to the next point, what is the difference between plasmid and a genome, or can a plasmid be considered as a genome or not? No, plasmid cannot be considered as a genome. Why? Because plasmids can mobilize. I just now showed that plasmids can be transferred from one particular bacteria to the other bacteria, but genome cannot be transferred from one bacteria to another bacteria. That means mobilization is possible in case of bacterial plasmid, but it is not possible in case of the genome. That is why this is the first reason because of which plasmid cannot be considered as a genome. Next important thing is that plasmids can be present inside a bacteria and also cannot be present inside the bacteria. Okay, it is not essential, but genome is always essential and it will always be present inside the bacteria. Next important thing is it is not indispensable. What does that mean? It is not at all essential for the survival of the bacteria. Bacteria can let go of the plasmid and also survive. That is called as indispensable. Okay, so it is not indispensable means that without it survival cannot be possible, but plasmid is not indispensable. That means without it survival is possible. I hope this is very clear in your mind. Now let us move forward and talk about plasmid size and shape. What about plasmid size and shape? Now understand this, that the plasmids can vary enormously in size. There is a large variation when it comes to plasmid. Large variation in sizes can be seen. So is there any general range which we can use in order to understand the size of the plasmid? Yes. So general size is around 1 to 200 kilo base pair. Okay, generally the size is around 1 to 200 kilo base pair. The next thing is F plasmid of E. coli. Now I'm telling you about F plasmid. The F plasmid of E. coli is having around 1% of the size of the E. coli chromosome. What does this mean? Let me show you with the help of an example. You might be knowing about F plasmid. So F plasmid means fertility plasmid. Fertility plasmid. F plasmid means fertility plasmid. Okay, now this F plasmid is around 1% of the size of the E. coli chromosome. That means if you consider the E. coli chromosome of 100 kbp, kilobase pair, then the size of the F plasmid would be just 1 kbp. I gave you this example in order to make you understand that what is the difference in sizes between the plasmid and the genome of a bacteria. There is a big difference. Okay, next, coming to the multi-copy plasmids have much smaller size. Okay, let me just move it up. Multi-copy plasmids have much smaller size. Now this is evident, right? Suppose there is one bacteria. Okay, suppose there is one bacteria. Now inside the bacteria plasmids are present. Now plasmids can be multi-copied and also they can be single copied. Okay, plasmids can be multi-copied also and single copied also. What does multi-copied means? Just simple, right? So whenever the copies of a particular plasmids are more than one, they are called as multi-copy plasmids. Suppose a plasmid has 200 copies, 300 copies inside a bacteria. Now if it has such a high copy number, then definitely it should have a smaller size. Why? Because if it has a larger size, it cannot be incorporated or accommodated inside the bacteria, because bacteria itself is of a very small size. You cannot incorporate or accommodate very large size plasmids in high copy numbers, right? So whenever there is a large size plasmid, it definitely would be present in very small copy numbers, and whenever the size of the plasmid is very low, it can be present in greater copy numbers. I believe you would have understood up till now. Let us move forward and talk about the next important point, that is, I have given you one more example, that is Col E plasmid. Now you see, Col E plasmid is further smaller than the F plasmid also. It has the 10% size of the F plasmid. That means it is further reduced. It has a further reduced size if you compare with the F plasmid, 10% of F plasmid, right? I just told you that 1% of the genome was F plasmid and 10% of F plasmid is Col E plasmid. I believe you would have understood this. Now initially I gave you important point that in case of eukaryotes, yeast has a 2 micrometer plasmid, right? This was the size of the plasmid present inside the yeast. Okay, next important thing is, I also told you that the majority of the plasmids are circular in shape and double-stranded DNA molecules. They are DNA, double-stranded DNA basically, and are circular in nature. But I also told you that variations are possible. So I have given you one variation over here, that some plasmids can be linear double-stranded DNA molecules. That means everything remains the same, just circular changes into linear. How can that be depicted, or what would be the shape? So you can see there are two bacteria that I have represented over here, Borrelia and Streptomyces. Okay, so you can see over here the bacteria, these two bacteria are having linear, they do not have circular shaped plasmids, they have linear shaped plasmids. So not only bacteria, linear shaped plasmids have been seen in, apart from bacteria, they have also been seen in fungi and higher plants. Okay, you would have understood this much. Let us move forward on to the next point. The next thing that comes in mind is that only DNA plasmids are seen, or can there be RNA plasmids also? So definitely there are RNA plasmids also, and both single-stranded RNA as well as double-stranded RNA plasmids have been seen, and they have been seen in plants, fungi and animals also. Now coming to the last point of this particular lecture, that how many different DNA plasmids can be seen. Now if you run a DNA plasmid, what I want to say is, if you have a DNA plasmids, if you have multiple DNA plasmids, you can characterize them on the basis of electrophoretic mobility. What does electrophoretic mobility means? In the presence of electric field, the DNA plasmid would start to move. Why will it move? Because of the negative phosphate group present. Now because of the negative phosphate group, this would be attracted towards the positive end. If it is attracted towards the positive end, it would show some mobility, isn't it? It is moving. Okay, if it is moving, understand this, that if it is moving, then you can divide it on the basis of the mobility. That means there are 5 possible conformations on the basis of the mobility, electrophoretic mobility. You can have 5 possible conformations of a DNA plasmid. Let us understand what are the configurations. So the first thing is, and we are talking about the electrophoretic mobility, and this is from slowest to the fastest. This would be the slowest and this would be the fastest. Okay, so let us talk about these. So the slowest would be open circular DNA with one strand nicked. What does that mean? Open circular DNA with one strand nicked, like this. Okay, so this is circular DNA. This was a circular DNA, but one strand has been nicked. So this looks like this basically. Relaxed covalently closed circular DNA, like this. Okay, linear DNA, like this. Fully intact supercoiled DNA, like this. Okay, supercoiled denatured DNA with unpaired region, like this. Suppose I'm drawing it like this. Okay, what does this mean? Supercoiled, this is supercoiling, I'm showing you, but this is unpaired region. That means it does not have any pairing over here. This is the unpaired region. So supercoiled denatured DNA with unpaired region, this is like this. Fully intact supercoiled DNA, this is the supercoiled DNA, it would be like this. This would be the linear DNA. Relaxed covalently closed circular DNA, and relaxed means it does not have any supercoiling, remember, right? And the final, the slowest one would be open circular DNA with one strand nicked. So this would move in the slowest fashion, this would move in the fastest fashion if you are running a gel. So basically, depending upon the electrophoretic mobility, we have categorized it into 5 possible the configurations. Okay, I believe you would have understood this. So this was the last thing that I wanted to talk about today. I would also want you to tell that we are starting a new batch. I've already made a video on that. The batch would be for GAT biotechnology students. We are starting that from 20th of July. So for those students who are interested for preparing for GAT biotechnology, you can enroll it. The batch is starting from 20th of July. So you can enroll in the batch and study with us and boost up your preparations. Okay, so if you want to know about the details of the course, you can call me up on 8299134528. So this is the number that you can call me upon and we can discuss further. Okay, so if you have liked the video, just hit the like button, subscribe to my channel, and don't forget to press the bell icon so that you can get the latest updates. Thank you so much and have a good day.